And next, I'd like to bring up Richard Finkel from Division Chief of Neurology at Nemours Children's Health System, who we're going to hear about natural history studies and early phase clinical trials.
Good morning. Let me start by thanking the organizers of this, I think, important and timely workshop on gene therapy. I was asked to speak about natural history studies for neurodegenerative diseases. I'm a pediatric neurologist, and my work is focused on children with genetic diseases, particularly neuromuscular conditions, which I'll highlight today. These are my disclosures. I do get a lot of grant support, some of which is listed here. I serve in an advisory capacity as well. I'm going to try to talk in the next few minutes about 4 topics that are listed here: the pediatric population, and I think to further on what you heard from Dr. Hai initially, and some of the ethical considerations from Dr. Kimmelman, and then to focus a bit on what is the true natural history or the unnatural history, because we're treating patients. It's not a true natural history, but we have to consider the world that we live in and the patients that we're treating in the clinic, and importantly, the impact of standard of care. And then I'm going to talk on clinical trial readiness issues and how it's, I think, important to the design of efficient clinical trials. So to start with the pediatric population, it's obvious to this group that the fetus, the newborn, the infant, child, and adolescent, they're not the same. So when we talk about pediatrics, we really have to be specific, and there are differences as listed here that are really fundamental to a clinical trial, such as what's the blood or CSF volume that relates to drug delivery or target engagement, drug metabolism and excretion, such as affecting drug exposure and the safety profile. And important for gene therapy, if it's going to be weight-based per kilo, you start getting into rather large doses if it's given systemically, as we're starting to experience. And then with intrathecal administration, there may be a fixed dose, but again, there may be differences in CSF volume or in the actual tissue for target engagement. And then I think what was discussed a bit earlier by Dr. Ha was, well, we're looking at a particular population that we want to study, but the pediatric population may be different from that in adults. And we may need different outcome measures. We may need a different study design. So it's not quite as linear as we would hope. And you could look at an example like juvenile Huntington's disease. And it's an early expression, but it presents differently. And you can't really use the same outcome measures in adults that's currently undergoing clinical trials as you might in a pediatric population. And then finally, I think we have to consider that the off-target effects of drugs may well be different in the growing child. So to the point that was brought up earlier, where do we start? So if the disease in question is present in adults, are we obligated to start in the adult population, in part for ethical reasons, because over age 18, you can presumably give informed consent rather than the parent or caregiver giving the assent for that child? Excuse me. So do we really need to start in adults? Well, you heard a convincing story from Dr. Hythe to say, no, I think in certain situations you can be persuasive that if the disease and the impact and if we know that early treatment makes a difference, that you can in fact start in children. But what about infants? Where do we actually start? And if we're trying to capture the most robust response to a drug, it may be in infants soon after the onset of symptoms. So we're struggling with those topics. So let me give 2 examples. First, I want to just mention Duchenne muscular dystrophy. And what's shown here is from an old textbook of a boy with DMD. And what has not changed in the 100 years or so since this photograph was done was that the incidence is still 1 in 3,500 boys are born with Duchenne muscular dystrophy. They present with symptoms typically between 2 and 4 years of age. What has changed is that the loss of ambulation, which historically was around 10 years of age, meaning it was a progressive disease, progressive weakness. These boys came off their feet, went in the wheelchair around 10 years of age, and there was a small variation between about 7 and 12 years. But no one was walking after their 13th birthday. That was the definition. That has changed. And that's changed for two reasons. We've learned that steroids promote longer ambulation and extend it by about 3 years or so. So if you look here, you'll see that in the red on this Kaplan-Meier, and this is a loss of ambulation Kaplan-Meier curve, that about half the boys have lost independent ambulation now at about 13 years. So that's changed from 10 to 13 years. And importantly, we've also learned that the type of mutation you have in the DMD gene makes a big difference. So if you have a mutation that is skippable by exon 8 using a skipping strategy, you can see that those boys actually do quite well. You can see that very few of them actually lose ambulation up to age 20, whereas, again, the red represents the overall cohort of boys. And if you look at the blue here, which is the exon 51 skippable population, you can see that it's quite different. So now, when we think about clinical trial design, we really need to consider how well do we understand the phenotype based upon the specific genotype for that population. So, for example, if we're doing a drug that is skipping exon 51, well, we need to very well understand the natural history of that particular group. And then how generalizable is that information for the whole group of boys with DMD? I'm going to turn now to spinal muscular atrophy, which is something that I've focused on the past several years. And I'm just going to briefly review the genetics for those of you that may not be familiar with this. So there are two genes, the SMN1 gene and the SMN2, what we'll call the backup gene. And this was identified by Melke and colleagues in 1995. And what was identified is that the SMN1 gene, as shown here, has these 8 exons or coding regions, and that there's the backup gene, the SMN2 gene, which due to a single nucleotide difference is alternately spliced, and about 90% of the transcripts fail to include exon 7, and thereby create an unstable protein. So the situation for a little child with SMA looks like this, because there's the absence of a functional copy of the SMN1 gene, and those children are totally dependent on this backup gene, which normally you don't need. If you have the SMN1 gene, you don't worry about SMN2. But in the case of the child with SMA, they're totally dependent on this little bit of protein that's generated from this full-length transcript. Okay. So there are two treatment strategies. One would be replace the missing copy of the SMN1 gene. That's the gene replacement therapy. That's the Avexis 101, and which was approved recently as Zolgensma by Avexis. The other treatment strategy looks at modifying the splicing of the SMN2 gene to try to promote more of the full-length transcript and thereby make more of the full-length protein, because this protein is identical to that that's made from the SMN1 gene. Okay, and this is exactly the way that nusinersen works, and also a drug that's in clinical development called risdiplam. So we have this drug on the market, the Spinraza, it's called. Okay, and this is important because when we talk about the clinical trial design, it's, well, can you do a randomized controlled study when there is a drug that's approved that's now the gold standard? Okay, so the history here is that first to the market was the nusinersen, or Spinraza, and then about 2.5 years later, we get the gene replacement therapy. So I'm going to run through a little bit of some of the issues we were facing in trying to design these clinical trials, and you'll hear more from Dr. Kaufman shortly. Okay, so what about clinical trial readiness? So shortly after the gene was identified, scientists were trying to develop animal models, but there is no naturally occurring animal with SMA. So they developed a knockout with and they put in the SMN2 transgene. So you had a very informative a severe mouse model, which we call the Delta 7 model, developed by Arthur Burgess and his colleagues. And there were other models that we learned a lot about the biology, but weren't actually so useful in trying to test for drug. And then more recently, there's a knockdown model in pigs. So there are different models that have served different purposes. And we were very fortunate that this this transgenic model, what we call the delta 7 mouse, actually did very well recapitulate the human condition and did predict response to drug. And as you know, that's often rare that you have such informative animal models. Now, at the same time that the neuroscientists were working on this side of things, in the clinic, we were working on the clinical side. So let me just show you what was being done. So this is work by Marco Pacini that looked at an ASO, in this delta 7 model. So he was showing that there was some restoration at the neuromuscular junction, which is faulty in SMA, restoration of the muscle, improvement in grip strength. So at the same time, we're trying to struggle with what do we do in the clinic to try to understand the disease better, because we saw that these drugs were coming fast and furious, but we weren't ready really to address them yet. So we then go into the clinic and we say, well, we need to learn more about the disease. So the first thing was to understand that the number of copies of this backup SMN2 gene is inversely proportional to the severity of the phenotype. So the more copies you have, the milder the disease. And then we took it a step further, and we developed a study group, and we just looked at the babies with the severe form of SMA called type 1 or Verdner-Kaufmann disease, and we saw that if you look at the babies that have 2 copies of this backup gene, the survival curve, again, Kaplan-Meier survival curve, is very different if you have 2 copies versus 3 copies. And we thought that a clinical trial design should focus on the babies with 2 copies because it's more predictable. When this was presented to the FDA, however, they felt that, yes, that's very striking and important, but it's also very dependent on standard of care. So there were a lot of discussions with the FDA on how to construct a proper clinical trial. So moving forward, we had to develop outcome measures, and we developed these motor function scales, and these are surrogates for strength or weakness. And we found that one size did not fit all, that we needed different scales for the infants than from the older children or even adults. And we also developed electrophysiologic measures and biomarkers such as a neurofilament that can serve as both a predictive and a prognostic biomarker to predict disease course in the untreated patients as well as response to drug. And this is ongoing work. I think what we also learned through these natural history studies was that age is an important variable. And shown here is work from an international collaborative effort to look at about 1,000 children with SMA. And what I want to point out is you can see the large variability in a change in a motor function scale over 1 year. That's what's shown here. And this is 0, and you can see that the young children between 2 and 5 years of age, some went up a lot, some went down a lot. And as they got older, there was less and less variability over 1 year. This is an untreated population. And if you look in the right-hand panel, I think you can see that in blue are the non-ambulant or the type 2 children with SMA. And they actually don't change very much here in the under 5 age group. But look at the large variation in the ambulant population. So this speaks towards the need to really understand your population that you're going to study and to notice that certain subgroups are more amenable to change without drug. So when studies were done, such as with valproic acid, and we saw some improvement in the younger children, we thought, oh, the drug works in younger children. When in fact, no, what we were seeing was that the natural history was demonstrating itself here. So I want to just spend a minute on the importance of standard of care. This slide on the left, the figure shows that there was a marked improvement in survival on this Kaplan-Meier curve from the 1980s into the 1990s. This was without a drug. This was showing improvement due to better standard of care, giving non-invasive ventilation support, giving nutritional support via feeding tubes and the such. So obviously, this is a huge response, and we need to take that into consideration in these clinical trials. That needs to be built in. Now, I want to talk about a gene therapy study, which is a phase 3 study. This is the gene therapy of Exus 101, and it's in the little babies with the severe form of SMA. And there was already this Spinraza out on the market. The parents had a choice of that. So we really couldn't ethically do a randomized controlled study with a sham control group, as had been done initially in the nusinersen clinical trials. Rather, we used this historical control, which I showed you earlier. This is the babies with 2 copies of the SMN2 gene. And in discussion with the FDA, it was clear that they wanted to see a very robust response if you're going to use a natural history control. Fortunately, that was demonstrated here. And this is an ongoing study, but you can see again, on this Kaplan-Meier curve, the remarkable survival in the treated group in red versus the historic controls. In addition, we saw that those same children who were treated early after diagnosis and after symptom onset seemed to respond better, and that most of them actually eclipsed this 40 points on this scale. And we learned from our natural history studies that no babies got above 40 points. So what we've learned is that, as you heard earlier, early treatment makes a difference, and we really need to understand the natural history to be able to put this into proper context. So to finish up, I learned a lot. I was privileged to go with some of the sponsors to the FDA and get involved in some of the early discussions. And I think the advice to go early and talk often really paid off with both of these drugs. And there's a nice paper that I'm showing here from members of the FDA. And it's really worth reading because I think it gives you their perspective on things. So with that, I want to summarize that I believe that pediatric studies have particular challenges and regulatory requirements, that understanding the nuances of the genotype-phenotype associations can really help design an efficient clinical trial design, and that provision of standard care is necessary to minimize both patient variation, but also to understand that it adds a second treatment variable. So with that, I'm going to stop. I'm going to just acknowledge the many patients and parents who participated in the studies that I've highlighted here just very briefly, the many colleagues in our study group, our funding sources, and particularly the patient advocacy groups that are listed here in my institution, Nemours Children's Hospital in Orlando. Thank you very much for your attention.